and I'm just going to update, there's some funding and conflicts. So this is the meeting. It was in 2014. It was at MD Anderson. It was an 8 p.m. meeting. And you can see Bob is there. Many of the people, some who are here in this audience here, were there. And I found my old slides. I keep everything on my laptop. And these, I did not modify the bullets at all. All I did was change the title. So this is, we wanted to appreciate the field of imaging genomics. Why? To aid in discovery with our colleagues from biology, to develop predictive models with our clinical colleagues, to ensure the physical image quality, standardization, harmonization, and to enable the use of image-based phenotypes and big data. And at this point, this was APM sponsored. So to understand the role of APM and in this venture, and to do strategic planning. And then we had many talks and discussions and breakout groups. And we have a white paper. And I'm just going to update, there's some funding and conflicts. So this is the meeting. It was in 2014. It was at MD Anderson. It was an 8 p.m. meeting. And you can see Bob is there. Many of the people, some who are here in this audience here, were there. And I found my old slides. I keep everything on my laptop. And these, I did not modify the bullets at all. All I did was change the title. So this is, we wanted to appreciate the field of imaging genomics. Why? To aid in discovery with our colleagues from biology, to develop predictive models with our clinical colleagues, to ensure the physical image quality, standardization, harmonization, and to enable the use of image-based phenotypes and big data. And at this point, this was APM sponsored. So to understand the role of APM and in this venture, and to do strategic planning. And then we had many talks and discussions and breakout groups. And we have a white paper presented in some newsletter on this. So if we think about it, this is what we're doing today. And why do we have AI in medical imaging? So most of my work, not all of it, some of the stuff is with special imaging acquisitions or microscopy, but the bulk is on images that are clinically and routine obtained in the population. And we want to ask questions about the relationship between features seen in the image and the biology of disease. And this is when I get to the two groups. One is to detect and diagnose disease early, and the other is to give the right patient the right treatment at the right time. And I've been realizing this. So the detect and diagnose disease early, that AI in medical imaging, you can go back as far as 1960s. It really took off in the mid 1980s. And that's the CAD group. I'll just use that term to make it easy for folks to remember. And there, our goal, we were filtering images. We were bringing out features of the images, as speculated or not, to find diseases, specifically in lung cancer and breast cancer. And then along the way, the people in more therapeutic wanted to give the right patient the right treatment at the right time. And I think that's where radiomics came in to look at how do we predict who's going to do well under treatment or not. And over time, these groups have come together, which is awesome. They worked in parallel for a while. And I know they worked in parallel because when I talked to some of those old timers from radiomics, and I mentioned the old timers from CAD, they didn't know each other. But the good part is we're all working together now. So here, there's a lot of tasks that you can use it for AI. I specifically focused on quantitative image analysis, CAD, radiomics, machine learning, whatever you want to call it. And there's many interpretation tasks. So let's go back to the group. CAD was to help radiologists to look at things that you could see in the image, but might have been missed, oversight error. Whereas for treatment, it was, can we characterize it on stuff that we can't really see? And in between, we had folks working on prognosis. So there's many tasks. Back in the 90s, we looked at radiographic texture analysis for looking at the density and the structure of the parenchymal patterns in digital mammograms. And that method, our RTA workstation, we now use across multiple modalities and disease types. And in the 80s and 90s is when we started with the computer vision and machine learning for lesion detection. This is screening, this is detection, diagnosis. And that was translated and enhanced by our two technology, went through FDA and all. Right, it needed improvement. We learned patients wanted AI in their images. And now, of course, it's at a much higher, that was 20 years ago. So now CAD is at a very high rate. And now a lot of the work we do is on diagnosis, prognosis, assessing treatment response, and disease discovery. So what I'd like to point out, and this is how we view our research, is when you develop AI in medical imaging, you can't do it in a vacuum. You have to look at all these. You have to look at the image formation, the physics of it, how the image was obtained, because it might be different between system A and system B. You have to recognize garbage in, garbage out. Make sure that your data is diverse, representative, and curated. And that's many people have noted that. And then also how we're going to harmonize it. Developing the AI algorithm. Is it human engineered? Such as, are you pulling out characteristics of size, shape, speculation, texture, or is it more a deep learning approach? I suggest you always look at both. And explainability, interpretability comes along. And the important thing is to know your end user. And then also how to assess it. What is the correct metric to assess it? And are you careful about your training, validation, and testing? Is it by case and not by slice? And also bringing in what is a sequestered data set. And on this last part over here, I have how is AI used? Initially, it was as an aid to the radiologist, computer-aided detection, computer-aided diagnosis. It was a secondary or concurrent reader. And one of the reasons it didn't do so well in the very beginning was because if you have a system that takes in type A images for type B task, and someone puts in type X images and it doesn't work, you know what they say? AI didn't work. So one, you have to know who your end user is. They have to know what are they inputting and what is the system, what is the claim of the system. So it's really a multidisciplinary effort. This is some of our early work. We called it a virtual biopsy. We used fuzzy C-means to segment the lesion. And then we pulled out various lesion features, used to call them mathematical descriptors, call them radiomics. They're extracted from the lesion. And then you can merge them with a classifier to yield a signature. If you train on cancers and non-cancers, it's a diagnostic signature. If you use different cancer subtypes, say on aggressiveness, you can train to give a likelihood of aggressiveness. And we called this a virtual biopsy because it was automatic, it was 3D, basically non-invasive. This is DCE MRI, so not totally non-invasive. Covers the complete tumor and it's repeatable. And here's just one of the features. And I put this up because I think we're all, like I said, working together towards this similar thing. This is showing intratumor heterogeneity. This is a 4D approach, 3D over time, breast DCE MRI. It's in 2D for the ease of, is this a pointer? Yeah. So ease of showing. These are all the voxels over time showing the uptake. And you see there's a lot of variation. And what we were interested in is two things: pulling off the most enhancing voxel. So we did fuzzy C-means clustering again. We got the most enhancing curve. And from that, we pulled off of the kinetics. From that, we also had, you can see here, the heterogeneity of those most enhancing voxels. So we did 3D texture analysis to give us an enhancement heterogeneity feature. And then we also looked at the volume of those most enhancing features. And we found that these different radiomic features were useful for different tasks. One might be better in cancer versus non-cancer. The other one might be useful in assessing risk of recurrence. Okay, we then moved to deep transfer learning, where we realized we didn't have enough cases. So we didn't want to train from scratch. So we used transfer learning. No lesion segmentation required, no extraction of features. We trained on networks that have been trained with ImageNet, cats and dogs, cars and all. And then we pulled off the features, or sometimes we did fine tuning, but the same question: can we distinguish between cancers or not? What we found, and shown here, we have the likelihood of cancer from human engineered. You can think of the emerging radiomic features, and here from deep learning, we found in a perfect world, all the reds, cancers would be here and all the greens would be here, but it's not that way. These are example cases. If it's outlined in red, it's cancer, outlined in green, it's benign. Sometimes the human engineered radiomic approach did better than deep learning and the other way around. So we looked at merging them for a mammogram, ultrasound, and MRI, and found when we merge them, we did significantly better. So we continue to always look at both of those. We've moved along with this where we looked at multi-parametric MRI, and we pulled off radiomic features from each of the different types. And we wanted to ask the question, how do we merge all these? The human eye-brain system is looking at all these from a radiologist. How do we merge the features and then go through a classifier, or do we merge the outputs of each classifier, which is giving a likelihood of cancer? And in both times we did that, we did show that either fusion method improved significantly on just DCE alone. We did this with deep learning, but now we had the added benefit of also merging in the feature domain. So we can add in the merge in the feature domain, in the classifier domain, or not just here we merged in the image domain. So we had 3 different ways of merging. And again, we showed, as expected, that we did better by doing that. So this is merging of radiomics with deep learning to see where we can go. This is that feature looking at the most enhancing tumor volume, but from the fuzzy c-means within the lesion. And we showed that it was very useful in predicting recurrence-free survival. This was on the I-SPY 1 data set, and we're now working with the I-SPY 2 data set that's become available to see how it performs. We did find that the performance varies with the different subtypes, which was also expected. And here is with imaging genomics, where we now have our various tumor characteristics, shape, size. This is the heterogeneity of uptake that I talked about, and we related to different pathways. This was with the TCGA, TCIA breast MRI mapping project. And here what we found, and we of course worked with, there are various biologists involved in this, and that we were able to relate because we found correlation, we wanted to make sure it also made sense, was here that this heterogeneity here was probably related to the heterogeneity of angiogenesis within the tumor. And along the way, we also recognize, if you, what is the repeatability of the computer if you have it run again based on, say, a different set of test sets, training sets, I mean. Basically, what we're after here is a computer would say, this is 50%, let's say 80% likely it's cancer, and I'm only 50% sure. So we're trying to come up with a sureness metric that would help do that. And we've moved along. We do a lot of other work there. Here's some just examples of work going on in my lab, as well as methodology. But what I want to get to before, since I started late, I know I have like 3 minutes left, is MIDRC. MIDRC started in August of 2020. It started with a budget from NIDIB of $20 million. It's a collaboration of 20 institutions, over 100 investigators. It's a partnership between APM, ACR, and RSNA. It's hosted at the University of Chicago, and it has 2 major parts: an open discovery data commons, so a lot of infrastructure building, and a machine intelligence and development. So here currently we have ingested, and this is COVID money, this COVID driven. We focus on chest x-rays and CTs, and Paul Kinahan will talk about some of this also in his talk, I hope. And we've ingested over 152,000 imaging studies. So a CT scan and the corresponding, say, chest x-ray with it is 1 study. So we're not talking images, we're talking imaging studies. They are curated, and Paul will be talking about that. And currently we have 75,000 imaging studies open to the public, and go down and download them. The website for MIDRC is given here, midric.org. And basically we have 2 inputs, RSNA and ACR. The images come in, they go through curation, de-identification, all. They are hosted at the University of Chicago on the Gen 3 Data Commons platform. That's data.midric.org. 80% go to the public commons, and 20% goes to the sequestered that I'm going to talk about in a second. We recognize the need to collect diverse imaging data. We have contributions from 23 states in the pipeline, and we keep account at multiple intervals how we are doing for diversity. And we want to do this because we want to develop ethical and trustworthy machine learning algorithms. So we also have a working group that looks at understanding biases and how we might mitigate them. So that's the scientific aspect of it. This sequestered data commons of only 20% of everything we collect is restricted. No one else can touch it. It is to test finalized algorithms. If an algorithm wants to go through FDA, we have FDA involved in this to accelerate regulatory clearance. It will act as a large test set. And we believe we're the only data repository that has this to allow for fair comparisons of algorithms. When we sequester, we match the 80 and the 20% in the characteristics. We just don't randomly take 20%. And then what we do with it then is for a developed algorithm, a specific claim, a specific clinical question, a specific population, we can then draw from the sequestered comments to evaluate it, give the score to the owner of that algorithm. They don't touch the data, and they can potentially take that to FDA and all. If they come back again, we will draw again and test them, but we do not tell them how the different cases do. We recognize the need for multi-omics AI. So MIDRC has interoperability efforts going on. We are sharing 3,000 patients with the N3, which has EHR data, and over 1,000 patients with the NHLBI PEDAL study. We're also working with Argonne on federated learning. Go to our website. We have a lot of resources. There's some MIDRC people in the room. There's code on GitHub. Of course, it has to increase. You can learn about how to access the data. And we have various tools like an interactive decision tree. We have a COVID-X challenge going on and efforts on annotations. If you go to RSNA, you can come to the exhibit and actually annotate data. We have a lot of outreach, and we have a seminar series tomorrow, but you're supposed to be here, so it is recorded. You can listen to it. Join our newsletter. You get monthly updates such as where we are by the numbers. So we support the medical imaging AI ML ecosystem with a curated, representative, trustworthy data, assessing diversity and mitigating bias. We're a community of collaboration, and we're lowering the barrier of access so everyone can have that data. And we're going to accelerate this with the sequestered data set by helping folks go through regulatory. Our third year is about to start. We're going to focus on long COVID. So we're going to go from CT and radiographs to MRI and ultrasound. We're going beyond the lung to cardiac and brain. We're going to continue to improve our interoperability and have a real live case sequestering. So thank you. And my lab are the fantastic people behind this and all the investigators at MIDRC. And I want to end. This is the last time I talked with Bob. We were on April 2021. We were on an AACR panel on artificial intelligence in cancer imaging. The two groups continue to merge. Thank you.